Hello everybody and welcome to our new live demo with Dr. Brian Haynes. As usual, if you have any questions, please use the question box in the side of the program and Brian will read them at the end of the live demo and will answer them. Brian? You there?
Yes, I'm here. Thank you.
Okay, it's all yours.
Thank you all for joining us this evening or this morning if you're in Asia. This demo and webinar is an introduction to a new product offering that we have, which is the 6-well plate, and it's intended for long-term culture and long-term growth. The BioFlux system is a best-in-class solution for cell-based assays using physiologically relevant conditions. I'm not going to spend too much time in an introduction to BioFlux, except just the basic operating principles. This talk is more focused on the application for a longer-term culture cell that we can use. Flexion, the root parent company, has had a track record of really innovative microfluidic technology that started with the BioFlux system, and then and that was for looking at live cell interactions and physiologically relevant models. And then the same technology, same root technology of microfluidics, was applied to the IonFlux system to do automated patch clamping, and that's been really a huge game changer in being able to do ligand-gated channels. And then more recently, we launched the IsoFlux system, which facilitates the capture of rare cells, particularly for circulating tumor cells, and then the ability to process those in settings like next-generation sequencing. These are a couple of the customers that we've had over the years. Anywhere from companies like Johnson & Johnson to universities like the University of Toronto have had a lot of success with our product line. The basics behind well plate microfluidics, this is the key portion for both the BioFlux, the IsoFlux, and the IonFlux, is the idea that we use a microfluidic platform or backbone. So in the case of the BioFlux, this is a standard 96-well sized plate. So we have 4 different plate sizes now: the 48-well plate, the 24-well plate, and now the 6-well plate that we're introducing today. And the idea is that on the inlet side, you would put your media, your cells of interest, or blood in some cases, and then the fluid flows from one side to the other using pneumatic pressure, using air pressure. And that's what really sets aside our system. A lot of other units, such as IBIDI, for example, the fluid comes in different lines, in different areas. With the BioFlux system, all of the fluidics are kept within a consumable, so that if you're working with something hazardous, something like blood, for example, or a biofilm, that your cleanup is simply, removing the plate, that all of the lines just perfuse air, that liquid never touches any permanent part of the instrument. In the case of the BioFlux, a microscope objective would sit underneath of this, and that would allow for imaging of the system. The basic operating principle is that you would put your media, your solution, into one side of the plate. You attach an interface which facilitates the air that comes and pushes from one side to the other, and then you run the sample. This is true for biofilm work. This is the same case for platelet work, for thrombosis work, and also for basic cell biology. Since I started with the company, there's been a desire and a need for doing longer cultures and longer cultures at higher shears. So and we've got 2 different shear styles that we do, 0 to 20 and 0 to 200. Typically, for these longer term shears, people want dynes at around 5 to 10 dynes and want to be able to do this for more than a couple hours. And this is an example of a paper that came out of Developmental Cell back in 2012. This is out from Cornell Medical School. Now, if you see the top left column here, this is no shear. These are HUVEC cells, the endothelial cells, that are cultured in the absence of any shear flow. And then the next panel over, these are cells that are grown in the presence of shear. This is 12 hours at 10 dynes. And you see this alignment that takes place here, where not only do you have things, for example, in 15 minutes with shear, you've got changes in AKT. Given an hour or two, you've got other post-translational modifications. NF-kappa B moves in about 1 hour. Your message changes probably in a few hours after that, but to actually get cytoskeletal changes, phenotypic changes that you see on the surface of the cell, it takes usually over 5 hours. In this case, it was 12 hours at 10 dynes. This required the researcher to change the media out several times, but it facilitated these really incredible images that they took with a confocal. Now, after shear, they, in addition to that, they looked at knocking down this protein called S1P1, and it's just using short hairpin. But you see that by knocking this down, you eliminate the alignment, and that was the purpose of this paper, was whether this protein was essential for alignment, and then they rescued the alignment with this compound called FTY, which was to mimic the effect of this S1P1. The typical channel characteristics of a plate involve an inlet, serpentine, which is basically like a pressure resistor. It allows for increased travel of the flow so that you've got better control over the flow, and then an outlet. And between that, you have a viewing channel where the microscope comes in and images. In the case of the 48 well, you've got two channels that are ganged up together here. This is all done on the bottom of the plate is at number 1.5 cover slip, or 170 micrometers. Most microscopes are configured to fit that size. And if you've got an adjustable collar, you can adjust it just to 0.17. But most fixed objectives are set to this specifically. In the case of the 24 well plate, we have two inlets, and this is intended to do liquid handling. Two inlets and one outlet. So you have a little longer of a runtime here because the volume's higher, but you still have to change the volume depending on how long and how fast you want to flow. This is an image of the 6-well plate architecture. This isn't my blood. This is just food coloring. But this was perfused into, actually it's perfused into the outlet and pushed to the inlet. They can go either way. But you see the length of the serpentine, the viewing channel, and then of course the volumes are remarkably, they're identical. Given it illustrates the consistency of this consumable. In comparing the plate types, the first plate type that came out was a 48 well plate, and then shortly after was a 24 well plate. The number of assays you can run with a 48 well plate are 24, and with a 6 well plate are 8. And of course, if you're doing 1-for-1 with the 24 well plate is 8, and with a 6 well plate is 3. If you want to put this on a quattro system, which really just means that you put splitters on this so that you can run 4 plates at one time, you can get 12 samples using the 6 well plate. So you can have these long runs, and by the end of it, you can have an N of 12 instead of an N of 3, so that you can look at your variables versus your controls. In that case, you could really have 4 groups if you wanted to do an N of 3. The total volume for these plates, with the 48 well plate, it's 1 milliliter. With the 24 well plate, it's 3.5. Now, there are 2 3.5 inlets, but there's only one 3.5 outlet. That's why I have an asterisk here. So even if you're if you want to run the two together, you can only run to the point where you filled up the outlet well. Otherwise, you'll overflow the system. With the 6-well plate, we're looking at 17.7 milliliters. So if you're running at 0.15 dynes, this is the rate that you would run for a biofilm. For the 48-well plate, you've got 80 hours, and that's usually sufficient. Now, this is with water at room temperature. If you run something at 37 degrees, this will be a little bit faster. Usually, this is sufficient for most biofilms, but there are things that take longer that you would like a longer time period. And then you've got 208 hours with the 24-well plate, and you've got close to 30 days with the 6-well plate. At a half dyne, that's modified. Running at 1 and 5 dynes are what people who are using typical cell biology, where they want to look at endothelial cells that have been exposed to shear for a prolonged period of time. In this case, you get 21 hours, roughly, with the 6-well plate. When you do this with, if you want to do this at 10 dyne, like the customer did before, you get 10 hours. And that would, basically be enough for the experiment. You could probably refill the media at one time, and that would be it. This other publication likely refilled the media several times. The new part numbers for this, you don't have to write them down, but they're here if you want for reference. There's a 6-well plate for the BioFlux 200, which would have the screws in it. 6-well plate for the BioFlux 1000. This is immediately available. This one should be available within, depending, a few weeks. An interface for the 6-well plate for the BioFlux 200, which would screw down into place. Interface for the 6-well plate for the BioFlux 1000, which will snap onto place. The opportunities for growing biofilms using 6-well plates, this came out from a desire from tuberculosis researchers wanting to be able to do longer-term cultures. And they said, we like the fact that you can do this for 72 hours without changing the media, but could you do it for 20 days without changing the media? And some mycobacteria are cultured for up to 8 weeks. Right now, there's minimal data using with tuberculosis flow cells and the effect of shear. This is probably because nothing's going to grow within the first couple days. Most of the studies that have been done with shear and flow cells have just been on adhesion. So really, they're just looking at the properties of tuberculosis to be able to adhere to like a monolayer of cells or a certain protein coating, to study some sort of virulence factor with the tuberculosis. But to actually culture this over time, there's very little that's actually happened on that. For environmental biofilms, I'm going to show you an example of this. You've got a very low nutrient media. For example, using water from the San Francisco Bay, that you might have a little bit of organic material in it. There's a little bit of salt in it, of course, but it's not like LB. So it's going to take a significant amount of time to culture, and the things that you're going to get out of it are going to be significantly different than if you put LB into it. And then also something like a urinary tract infection, where you would use urine, and urine is it has some things that the bacteria can metabolize, but it's going to take a long time in order to get that out. I can show you here an example of an oral biofilm that I did using LB. And this is one of the channels that I started with. Show you here. We'll load this image. This was for, I believe, 198 frames. And we can put a timestamp on this. Display, graphics. Elapsed time, all planes. Maybe something a little bigger that we can read. When I press stamp, and that would tell me how long this went for. So this went for about 2 days, 17 hours, 40 minutes. It could go for much longer. This was just to show the effect as it was coming along. This is an example of growing environmental biofilm from water. You start out by collecting water in your source. You want this aseptic, of course. You centrifuge down. This is something from the San Francisco Bay, which has a lot of material in it. If you're going off of something that's pretty clean, you're going to need more than 100 milliliters to find viable cultures at the bottom of it. What I like to do is spin this down enough that it doesn't kill the bacteria, but it pulls everything down to the bottom, and then filter out the media, or I'm sorry, the supernatant, which will then be used as a media, and then reconstitute the pellet with the filtered water. That would be used as the inoculum. Prime the channel for with anywhere from 200 microliters. This is actually quite arbitrary, for 2 dynes at 5 minutes. Inoculate the culture, and then incubate for 1 to 2 hours to make sure that the material there is able to adhere to the glass, and then top off the media and perfuse for 3 to 10 days. This is an example of water that was cultured from the bay. I believe this is Vibrio, but I'm not entirely sure. I have some other, there's a paramecium actually, if you want to see that. We can replay that real quickly. It sort of jumps into the right there. Yeah, we kind of missed him. He jumps, or not he, this paramecium, I've got to be politically correct about it, jumps into the frame just briefly. But you see things that you wouldn't if you're culturing with LB. And this took about 5 days to culture, and then this is actually live. So these bacteria have a tail on them, and they're on the surface of the glass. They're being exposed to shear. They're being exposed to flow going through, but they're not flying off. So they're actually gliding and sticking on the glass itself, and they're sort of moving. It's from one area to the other. So this is another example. We have a collaborator at the University of Michigan that we're going to do some tuberculosis work with, and hopefully we'll have some primary data for you in the next couple weeks to, by the end of Q1 in 2016. At that point, I'm happy to answer any questions, but this is pretty much what I have for this webinar. Actually, no, I take it back. I'm sorry. I was going to show you how to use the plate. I guess because it's 5 o'clock here, I got a little bit off. So we have a 6-well plate, and I'm just using water and beads, so I don't need any gloves with this. I'm going to take and prime this plate with about 200 microliters of media. The inlet of the plate is on the bottom, actually. So you've got one up top here, and I'm going to prime from using the wells on the bottom are actually the inlets. And when we update the software, this will be reflected in that. I'm using a modified 24-well interface. Our 6-well interfaces are in manufacturing right now and should be finished in a week or two. And we'll have a modification on the software as well. I'm going to set this to 2 dynes, and I'm going to perfuse it in these channels. Sorry, this way. This is a little less than 5 minutes, but I'm going to go ahead and stop this and add some beads and then put this on the scope so you can see what this looks like. We've already had perfusion from one side to the other. Just beads. It's nothing that's remotely toxic. And we can take a look here at the, I'm going to close out this. This was the oral biofilm. And we have the channel here at 10x. And I'm going to perfuse 2 dynes. Given this is going at 2 dynes a little fast, so I'll put it at 1 dyne and then actually put a frequency on it so that you can see what pulsatile flow looks like. These are the same beads lighting up with red fluorescence. If I slow this down to 0.5 dynes, Might make it even easier to see. And given how bright they are, we really don't need much more than probably 50 milliseconds of exposure time. And if you wanted to be precise, you could say, oh, these actually are red, so we have them red. The camera picks it up as black and white. So to show you on the screen here, I'm perfusing at 0.5 dynes with a frequency of 1 hertz. So it comes up and comes back down. We have a couple publications on that in with Patrick Hines' lab out of the University of Michigan. He's had some really exciting data with pulsed outflow and adhesion. But I'm going to stop that, and I'll be happy to take any questions. Otherwise, I really appreciate your
Thank you, Brian. If you guys have any questions, you can use the question box, as I mentioned.
participation.